Enzymes, like I said in the previous video, are the catalysts in living things. So they are proteins that are actually used to speed up these chemical reactions that take place in different cells. So enzymes are needed for almost all biological processes. The activation energy that would be necessary for many of these biological processes, whatever, couldn't actually come from inside the body. Okay, there's not enough energy contained inside the body in order to kickstart all of these reactions. So enzymes are used in place of that. Again, most enzymes are proteins. Okay, so this is where you talk about those 4 biological molecules of life and how important they are. And they allow for reactions only under controlled conditions. So this goes back to homeostasis, which we talked about in the last chapter, okay, and how important that is and how several different conditions have to be kept within a certain range. That's because that's the range that these enzymes work the best in, okay? So you're talking about pH in the blood and the cells. It's also talking about temperature. Okay, so there is actually there is kind of a fail-safe for if you have a fever in the majority of your body. There are different compounds that your cells will start to produce in order to protect the enzymes so that they don't get destroyed just because the temperature has gone up. That works really well in everything except the brain. The brain doesn't have those, okay, for various reasons, which we won't go into unless you want to talk about it. But that is why it's so important to keep your body temperature under 103 degrees. Once you get up to 103, you can actually start having brain damage. Okay, so it can start to damage the cells simply because they don't have that fail-safe in order to protect from higher temperatures like that. So disruptions in homeostasis can prevent these enzymes from functioning. And enzymes function best within that small range of conditions. Okay, so that's why whenever you have your blood work done, okay, they will give you, if you look at it, it gives you a range of the good, like the good range for that specific condition. And then it'll tell you if you fit within that range or if you're high or low or anything like that. So temperature and pH are the big ones when you talk about enzymes or proteins. They can actually break the hydrogen bonds. Okay, so if you remember back from the first section of this chapter, hydrogen bonds are incredibly important to a lot of different things. So we talked specifically about water, but hydrogen bonds make up most of the interactions within a protein. So they hold the protein together, and they can help the protein keep its shape. So if those hydrogen bonds get broken, it basically unfolds the protein, does what's called denaturizes the protein, or denatures the protein. So instead of having this very structured shape where everything is kind of folded in on itself, and it creates a very particular spot where these reactions can take place, now you've got just kind of loops and like a string of amino acids. So you don't have the shape, and then because you don't have the shape, you don't have the function. Okay, so this is showing you a normal protein. You can see all the spirals and the loops and how it all kind of connects back onto itself to create this very particular shape. But then when you denature the protein, it just turns back into that string of amino acids. Okay, but then some proteins you can actually renature. So if you just cool it down again or get the pH back within that range, it will actually renature. Okay, but not all of them actually can be easily renatured. Some won't ever be able to fold back again because the protein actually folds as it's being created. Okay, and that creates a very specific shape. And that's something that you can't get again afterwards. Some of them will actually need another protein to come in and help them refold. Okay, so repairing the proteins and getting function back after the temperature or pH gets outside of that range can be difficult and sometimes impossible. Okay, so that's why your body works so hard at keeping both your temperature and your pH within those ranges. So an enzyme structure allows only certain reactants to bind to the enzyme. Okay, so this is very particular, and the reactants that actually bind to an enzyme are known as substrates. Okay, and they bind to the enzyme specifically to undergo a chemical reaction. And they bind in an area called an active site. So generally, the active site is relatively small compared to the size of the protein itself. So it's basically, if you look at this, this is kind of a very simplified version of it, but you've got your two reactants, your two substrates up here, okay, and they're coming in to react on top of this enzyme. So the enzyme basically is constructed in such a way that it will kind of force the reactants into a specific shape and alignment so that it's easier for them to react. Okay, and all of that is facilitated by the active site. So this is known as the lock and key model. So it illustrates how the enzyme functions. So the substrates get brought together, and then the enzyme will catalyze this reaction. But it's a very specific lock and key. Okay, so if you've ever tried to stick the wrong lock, like if you ever try to unlock your house using your car key or start your car using your house key, you know that doesn't work at all. Okay, so the key is shaped in a very particular way that matches up with the lock. And that's how you can lock and unlock everything. Okay, so it's the same kind of way that enzymes work. So enzymes will bring these substrates together, shape them in a very particular way, and weaken their bonds. So it's easier for that reaction to take place. Okay? Well, that is backwards. So substrates come in and bind on the active site. Enzyme will kind of corral those two substrates together and make sure that they're shaped properly, and then kind of just stitch them together, or make it easy for them to stitch themselves together. And that releases the product. Okay, so the final product, what you wanted out of that reaction. Okay? All right, and that is it for this chapter.